Good morning. Today's topic is gram stain. Though gram stain is an important differential staining procedure which is used in our regular microbiology lab. On the basis of the reaction in the gram stain, bacteria can be divided into two major groups. One is gram positive and another one is gram negative. This method was developed in 1884 by Hans Christian Gram, who was born in 1853, who developed this technique while working with Karl in the morgue of the city hospital in Berlin. At that time, he had found that this staining method differentiates bacteria on the basis of their cell wall. After gram staining, gram-positive bacteria appear purple-violet in color and gram-negative bacteria appear pink in color. The gram differences in the gram stain arises because of the differences in the cell wall of gram-positive and gram-negative cell. This stain can differentiate bacteria because gram-positive cell have a thick layer of a peptidoglycan and a gram-negative cell has a thin layer of peptidoglycan that retains the primary stain. These crystal violets dissociates in aqueous solution into crystal violet ion and chloride ion. This ion penetrates the cell wall of both gram-positive and gram-negative cell. The crystal violet ion interacts with negatively charged components of bacterial cell and stains the cell purple in color. Iodide interacts with crystal violet ion and forms a large complex, and the complex is called CVI complex, that is crystal violet iodine complex, within the inner and outer layer of the cell. After that, when a decolorizer such as alcohol or acetone is added, it interacts with the lipids of the cell membrane. The CVI complexes are washed out from the gram-negative cell along with its outer membrane. In between, CVI complexes of gram-positive bacteria is remain trapped because of its multilayer nature of peptidoglycan. The decolorization step is critical and must be timed correctly. The crystal violet stain is removed from both gram-positive and negative cell if the decolorizing agent is left too long. After decolorization, the gram-positive cell remains purple and the gram-negative cell loses its purple color. After that, counter stain is added, which is positively charged safranin or basic fusion. Both gram-positive and gram-negative bacteria pick this counter stain color. After staining, gram-positive bacteria appear purple-violet in color, whereas gram-negative bacteria appear pink in color. The color differences in the gram stain arises because of its differences in the thick and thin peptidoglycan layer. Now, let's learn about the gram staining procedure. In the first step, the smear is stained with the basic dye, that is crystal violet, the primary stain. This is followed by the treatment with an iodine solution functioning as a mordant. At that time, crystal violet iodine complex is formed. The iodide increases the interaction between the cell and the dye so that the cell is retained more strong color. The smear is next decolorized by washing with ethanol or acetone. This step generates the differential aspects of the gram stain. Gram-positive bacteria appear crystal violet color, whereas gram-negative bacteria loses its crystal violet color and become colorless. Finally, the smear is counterstained with a simple dye. For counter stain, we can use safranin or basic fusion. Mainly safranin is used. Due to this, gram-negative bacteria appear pink or red in color. In this way, we can differentiate these two cells. So this is all about gram stain. Thank you for watching this video.